Hello, everybody. Welcome back to our last day of webinar. And now I'm going to invite Edward Dye for his presentation. So welcome, Edward. Thank you
Hello.
very much for being here with us today. Thank you for accepting our invitation. Edward has a Bachelor of Science degree from UCLA and has worked at various universities and biotechnology companies using fine surgical tools, where he's the product and project manager in the United States of America Office for Fine Science Tools, and he's here today to talk a little about his work. So thank you, Edward, again, and you may start your presentation anytime you want.
Great. And thank you for having me here. And I appreciate the opportunity to speak about our products that will hopefully help you not only find the right tools for your particular application, but also get you over that last step. You know, I mean, everybody already spends so much time and effort to get to that sample stage, and having the right instruments can make a huge difference in your experimental procedures. Briefly, as everybody can hopefully see in my slides here, this is the course overview of the presentation. I was going to briefly provide some information about fine science tools. Also discuss just briefly about the common procedures that we do encounter with Zebrafish. And then basically the bulk of the presentation will be about the product recommendations and what we offer. And then we'll wrap it up with some specializations that FST can offer to you with our surgical instruments, and then open up to any questions and answers at the end. Briefly, as far as myself, I do have a neuroscience degree from UCLA, where I was studying pre-Boston complex or rhythm respiration for breathing. And so after graduation, I went to New York University, and where I did some malaria vaccine development work in the Dr. Nuisance Weiss lab. From there, I transitioned my skill sets and went to drug and gene delivery at the University of California, San Francisco, primarily in a liposome lab, basically encapsulating various drugs targeting various cancer tumor models. Before going into biotech, where I was performing a lot of in vivo pharmacology type of work, doing a lot of screen drug candidates. Basically utilizing those skill sets, I was able to find a position here at Fine Science Tools that utilize all these kind of fine instruments. And now in my position here, even going into further topics like zebrafish and other entomology kind of type of work that use these kind of instruments. And so hopefully I can share some of my knowledge in the past and also try to make some product recommendation that might be useful for you as well. So about Fine Science Tools. We offer over 1,100 surgical instruments for research. And what separates us from the other companies is that we only carry high quality surgical instruments. So we're using European-made surgical instruments. We're using German steel, German craftsmanship, and impeccable quality control check in our Heidelberg office. We have over 48 years of experience and primarily used for research purposes. The quality is actually good enough for human use, but we just avoid the whole FDA regulation of paperwork involved and the amount of regulations involved with that. But as far as quality is concerned, it is quite suitable for all type of applications. The hand finishing and quality control is actually done in-house as well. And one of the unique features of us is we can actually do custom-made instruments on request, and I'll go into that in more detail at the end of the presentation. We carry over 1,100 instruments in stock, and each year we do offer a new catalog that shows these items in real size. That's one of the benefits of having our catalog, is our new catalog actually for 2023 here, as we're trying to go green and with our nature talk. And we actually offer them in actual sizes. So that's one of the benefits of having the physical catalog. You can actually see how big or small these instruments are. That's not quite easily attainable on web. And we have 100% price transparency as well. And most of them are made in Europe as far as the surgical instrument line, and the accessories might come from the USA manufacturers. And with 3 offices, we are able to ship worldwide. So again, we're using German steel and German craftsmanship for our products, and sourcing only the best quality material and skill sets to make these surgical instruments. We have support and customer support around the world, trying to address your needs and trying to get you the right instruments for your right application. And this is another important feature that separates us from the other companies, our German quality control department in Heidelberg that actually goes through all of our instruments to pass a secondary inspection before shipping it out worldwide. So we have an office located here in San Francisco, USA. We have our German office located in Heidelberg and our Vancouver office in Canada. And actually our Vancouver offices can ship worldwide. But we also do offer 35 resellers or distributors worldwide as well, as indicated with these black little dots, to cater to your needs, if that's easier for you to place orders with. And we broke down our product categories to at least 5 different sections here. Basically, there's the cutting section where we have scissors. If you have tougher material, we have a bone instrument line, and even some knives and scalpels. The other section we like to call some grasping is basically highlighting various forceps of fine tips, broader tips, to various hemostats. In the exploration and closure areas, we do have various probes and hooks, which are very useful for zebrafish studies, spatula, pins, and other retractors of vascular instruments. That'll probably be more for the animal sector, or in vivo mice sector. But we also have various accessories as well for animal surgical laboratory and instrument care as well. Something to be aware of is our student line instruments. And this is a product line that we actually expanded tremendously last year, adding over 100 instruments to this. And it's what we call an introductory student instrument line into our product category. It doesn't have the same hand finishing and the amount of steel in each of these instruments, but it does offer quantity discounts, and it's a good way to learn if this is the right instrument for your particular application, or if you're doing a training course and have a lot of students and you just want to at least give them exposure to these kind of instruments. It's basically an introductory line again, and it's a great way to even give to visiting postdocs or training students to see if this is the right instruments without the initial investment into that instrument. All right, so briefly, just going to some common procedures that we get common questions for about zebrafish studies. And basically, it's various cuts and caudal fins, spinal cord, abdominal cavity, even genotyping, and immobilization. So I'm going to basically go over product recommendations to address those various type of procedures that you might be currently doing. It's going to discuss various features and characteristics of each of these following product lines, and hopefully some of these might be of interest for you. So when we get down to the spring scissor section, these will basically be the finest tip and cutting edges for you to use for your studies. The majority of these are handcrafted by FST, as they are overworked in our Heidelberg office. And there's also 35 different articles and 70 different styles. And by styles, I'm meaning by curvature up, sideways, 45-degree angle, different texture, different thickness of the blades as well, or different cutting action. Alloy material will also be another factor to take into consideration when choosing your spring scissor. Serration. A serration is like a bread knife, giving you that gripping action when you cut. And so as you can see here, as far as our finest spring scissor with the Vanda style kind of handset, would give you a 2mm cutting edge. We do also offer a 2.5 millimeter cutting edge and a 3 millimeter cutting edge. So depending on your tolerance for that particular application, you can go by a 0.5 millimeter difference for your cutting needs. Just for a price point of view, if you're able to tolerate a 0.5 millimeter difference, the second spring scissor here is a $100 difference in comparison to the 2 millimeter. And so that might be a great way to at least start with. That might be suitable for your application, but with a beneficial price point as well. But all three of these will basically give you that first initial precise cut. We do go up in bigger size. And basically the Vanis 2-Bigen spring scissor, the one in the middle here, that would be basically your baseline at a 5 millimeter cutting edge. To the right of that, we do have that micro serration, which it gives you that better gripping action, especially when you're dealing with very slick samples, I would say, something that's very slippery. This micro serration is a great way to maintain that smooth, clean cut without slipping the sample away. We do offer a Moria line spring scissor. It's French made from the Moria brand, and it is actually a bit heavier and has a bit of a smoother cutting action. And so some people prefer that extra weight when they're using these kind of spring scissors. We also offer a titanium version, and it's highlighted in blue. This is great for working in saline conditions, acidic conditions, but also it's lightweight. And so you're able to maintain some of that strength of a spring scissor, but lightweight. So if you're doing a lot of procedures in a day, over the course of that day, a lighter spring scissor will be beneficial, not only for ergonomic purposes, but also the length, the longevity of your instruments in such saline acidic conditions. And if you're finding that your spring scissors are tending to blunt or dull easy, we would recommend a ceramic coated spring scissor. This is highlighted by our black spring scissor right here. And this extra coating gives it that extra strength. And so these will last much longer in consideration to the regular spring scissor line. And also, if you were to work under a scope and you have under a light, you don't have that glare effect as well. So that's something to take into consideration. And now as we move up onto the fine scissors or ring scissors handles, as you would say, again, we're addressing some very similar characteristics associated with the spring scissor, but just scaled up to a bigger size. And some of the other features we have here will be a left-handed, for all those left-handed out there, and large loops. And so this might be more ergonomic and beneficial for those looking for a left-handed or large loop scissors. As far as our finest ones of the ring scissors, we would go with extra fine bond. And we also have this black handled scissor. And this is our tough cut line. And basically these are an indicator that we do have serrations on one of the cutting edges. So like a bread knife, it actually grips as it cuts. And it's great for tougher material, especially like cartilages, and just holds up better and should last twice as long in comparison to the bond scissor. The hardened fine scissor is basically an extra process on the tips, and so the blades and the blades on these will last longer also in comparison to the fine scissors there to the right. But otherwise, a fine scissor sharp, we have 10 styles of those, and those will be your baseline. And so that would be your introductory line, I would say, to see if this is the right kind of scissor for your application. And again, if you wanted that tough cut serration feature, I would go with the fine scissor to the right of that one. The gold handle is actually an indicator that we have tungsten carbide at the tips. And so tungsten carbide is a harder alloy, and it's actually inlaid onto the tips and welded in before they start fine sharpening the blades. And so with that extra layer of alloy on the tips, uh, these scissors will give you nice clean cuts and will last twice as long in comparison to your regular bond scissor or fine iris scissor. So that's why people prefer these to give you nice cleaner cuts over a longer period of time. This version here actually has a combination of both the tungsten carbide and the tough cut, so it has that serration. And so usually these type of scissors will last 4 times as long than the regular fine iris scissors or bond scissors. And we have a ceramic coated version. So we're basically combining all 3 features of tungsten carbide. We have serrations and then ceramic, the black coating all over these scissors. And these are would be the top of the line scissors that would give you 6 times longevity over the regular fine iris scissors. Transitioning to fine forceps and basically manipulating your sample with these forceps. And so we do offer a line of Dumont forceps, and I will talk about the tips, again, the alloys, even serrations, micro blunted to help minimize perforation of your sample. And so one of the basic things we need to take into consideration with these fine forceps is the type of alloy to choose. There are 5 different main alloys we offer, where we start with titanium. And as far as hardness, as far as the tip, that would be the least hard in comparison to the other 4 alloys we offer. Also, there's a characteristic of magneticism, temperature resistance, if it's autoclavable, and some of the advantages to choosing this alloy. Tamoximal and inox are some other standard alloys we offer, and we would say inox would be your baseline alloy that people usually start with. The inox alloy number 5 would be the starting point. If you want to go softer, you would go to Damoxil. You want to go softer than that, you would go titanium. But if you wanted to go harder than inox, sorry. If you want to go harder than the inox, we would choose a Dumastar alloy. And beyond that, after that would be a carbon alloy. And so that would be your 5 ranges of different alloys to choose from. And each of them have their own characteristic of being magnetic, the temperature resistance, and autoclavable. After choosing the proper alloy, we have 3 different tip profiles. And so standard is usually what we would say is a 0.1 millimeter tip width, while biology tip will be half as fine, which would be a 0.05. And then finally the superfine, which would be even half of that, which would be a 0.025 millimeter tip size. And these will be the 3 different tip sizes we would offer for the various different alloys. And so as you can see, we're already doubling or tripling the amount of various Dumont forceps we can offer with the alloy and the tips dimension to choose from. So as we usually start with our catalog, we usually go with the finest, and this will be the 5SF, the number 5 superfine, which has that 0.025 millimeter tip width. We do have a version here where it's a biology tip, a 0.05 millimeter tip width, but this version actually has a feature that pressing on the handle, the tips won't go up in size, and so it actually stays flatter with additional pressure in the handles. And again, ceramic coated, a great way to have a fine biology tip forcep with stronger tips in comparison to the number 5 SF, and also the whole corrosion resistance, anti-glare, and just holding up better in comparison to the number 5 SF. This version here, the number 5 CO forceps, is basically one of our lightest forceps that we offer. And again, for ergonomic purposes, if you're going to be using this all day, eventually does add up. And so having this type of forcep with that fineness is a great benefit. And micro blunted. And so in addition to straight, we have at least 6 different tip profiles, whether it be 45, 90, 10 degree, like a curve, number 7 version of these micro blunted Dumont forceps. The benefit of these, they just take a little bit off the tip, and so it helps minimize the perforation that you might experience and damages to your tissue samples in comparison to that regular number 5. And the 55 is almost similar to the number 5, but people swear by these number 55, and the main difference is just the handle and the shank of the number 55. So for those dealing with sticky samples, we do offer a mirrored finish forceps, and at the tips it has that glass coating on it to help make your sample slide off a little bit easier. This number 5 is your basic standard, and that's why we had 9 different styles, offering all the different type of alloys and tip sizes of standard and biology. But again, this will be your baseline forcep to choose from to start with, I would say. And the Dumont WA number 5 is one of the new versions of the Dumont forceps. And it's basically, if you were to see a side profile, it's one singular piece folded together or bent together to come together, versus the one on the left where it's actually 2 pieces fused together to give you that V action. This is more of a U shape, and it's easier to clean, has a little bit more spring action, and it's just a different feel and weight to it in comparison to the typical number 5. Moving on to punches and cores, and those who were to use this for genotyping, we do have various punches to offer. And the finest, again, would be a 0.31 millimeter inner diameter, or ID, or an outer diameter of 0.61 millimeters. And we have the NIH style neuro punches, where you can actually punch and then you could blow out your sample through that, or we have a sample core where you have a plunger to actually expel the sample collected. Another feature is immobilization, keeping your sample pinned to your sample, to your working table. And so we do offer the minution pins of a 0.1 millimeter rod diameter that is also useful for your experimental procedures. And now coming to our FST workshop in Heidelberg. This is something unique where we can actually do custom orders, or we can actually procure items for you that is not readily available in our product catalog or online. For example, if there's like a spring scissor that you are interested in, but the tip thickness is just way too big, our German craftsmanship in that Heidelberg office can actually hone it down to your specification. If there's some kind of variation you would like, you need a 36-degree angle for a particular reason, our German office can probably do that for you as well. And so this is where custom-made tools for your particular application can be done by our German office. And this is where we would try to hold meetings and discussions to learn more about your experimental procedure and try to find you the right tools. As a person myself, being in a lab, I've tended to gravitate to what was in the drawer next to me and try to use that to adapt to my particular science studies. And so working at Feinstein's tools and seeing all these instruments and knowing that there is a particular instrument for a particular procedure is just a game changer, in my opinion. And even getting craftsmanship, quality craftsmanship, custom tools done as well is another game changer that can be taken advantage of, that not many people really know about from Feinstein's tools. And that will conclude my presentation today. And hopefully I can answer any questions that you might have, as I'm looking at the private chat here, hopefully in English as well. And at this time, I'm trying to see if there's any questions you might have. Also, my email address is here at edy@findscience.com. And so if you did have a questions you want to ask me later on, or if you want to talk to me later in more detail about custom orders, please do let me know.
Thank you, Edward, for your lecture. It was a great lecture. And I don't think we have any question. Yves, something from YouTube? I don't think so. So thank you very much for the presentation.
Absolutely. Thank you very much for having me here today.
And now we are going to wait the next one. Thank you.
All right.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community. And we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us. Because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries in research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps, and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
Hello, welcome back. For our last presentation of this webinar, we're kind of sad that it's the last one, but let's do it. So I'd like to invite Professor Patti to the conference, please. So Professor, thank you very much for being here with us today. Professor Pat's research is focused in the rapidly evolving field of metabolomics. His group is developing new mass spectrometry and nuclear magnetic resonance technologies by using state-of-the-art mass spectrometers coupled with cutting-edge metabolomic technologies. They take a system-level approach to study comprehensive metabolism and identify specific pathways that are altered in connection with particular phenotypes. They strive to translate their metabolomic findings into a physiological context through the use of classical biochemical tools and animals models, as well as with the development of new technologies, such as mass spectrometry-based metabolic imaging and whole cell nuclear magnetic resonance. So welcome, Professor Patti. Thank you very much for being here with us today.
Thank you. Can you hear me okay?
Yes, we can.
Perfect. Excellent. Well, thank you for the opportunity to present today. I'm going to be talking about exploiting some of the metabolomic technologies that were just mentioned in the context of zebrafish biology. Even though I have the pleasure to give the seminar today, I'd like to thank the person who has really invested all of the blood and sweat, who made all of this work possible. It's a senior, talented graduate student in the group, Maddie Jackstad. She has remarkable creativity. She has enormous perseverance and an amazing work ethic that has made all of the research that I'm about to present for you possible. My talk today is going to contain 3 parts. First, I'm going to provide a brief introduction to metabolomics. Second, I'm going to tell you about some different methods that we've customized specifically for the analysis of zebrafish. And then lastly, I'm going to speak a little bit about using metabolomics to understand metabolic physiology or organismal metabolism or systemic metabolism. So let's start here with the first item, and that is an introduction to metabolomics. So in my laboratory, the way in which we perform metabolomics, and I should mention that the goal of metabolomics is to quantitatively assess the small molecules or the metabolites that are present in a biological system, in this case, zebrafish. And the way in which we perform metabolomics in my laboratory, for the most part, is by using mass spectrometry, specifically liquid chromatography mass spectrometry. So I want to start here today by providing you with a little bit of background on the general workflow that we apply. The first step is sample preparation. So in the context of zebrafish, you want to isolate all of the metabolites, and that means getting rid of all of the macromolecules. This is going to be things like proteins and genetic material. After you've isolated the metabolites, then you will perform liquid chromatography mass spectrometry, and I'll abbreviate that with the acronym LCMS. When you analyze a biological system such as a zebrafish with LCMS-based metabolomics, you generate really large data sets, can be on the order of a gigabyte or even more. So it's really impractical to analyze those data sets manually. We rely on software for data processing. And the output of those software that we use for data processing is the production of somewhere in the vicinity of 20 to 50,000 different signals. And those are unique ions that have unique MORZ values and retention times in the data set. So just note here that you're getting a really large number of signals in the experiment. Now, of course, trying to interpret signals in the context of biology means that we have to be able to identify what biochemicals those signals correspond to. And that requires additional steps to encode to determine what those signals are structurally, to assign names like glucose or alanine or lactate or whatever your metabolite might be. You'll notice on this slide that the steps of the workflow that are on the top are colored green and the step on the bottom is colored red. And that is to indicate that it is widely recognized in metabolomics that the rate limiting step of doing experiments is really translating those signals into identifications and subsequently interpreting those metabolites in the context of biochemistry. So let me give you a little bit of a flavor about why it's challenging. Why does that lead to barriers in our ability to interpret metabolomics data? And to motivate that and to appreciate why it's challenging to interpret metabolomics data, I'd like to give you this graph here. What I'm presenting here is if you take a zebrafish and you can either analyze specific tissues in the zebrafish or analyze the zebrafish intact, actually doesn't have much of a big impact. What you end up detecting by metabolomics, LC-MS-based metabolomics, is on the order of about 25,000 unique signals. But what you can see in the graph here is that even though we are detecting 25,000 signals, that the fraction of those signals that we can successfully identify and assign a biochemical name to is relatively small. Just that little sliver there that's shown in blue that we're able to positively identify. So this raises the important question of what all of these other signals are. These 20,000, 20-something thousand signals that we cannot identify that we're detecting in a zebrafish, what do they correspond to? Now, your first knee-jerk reaction might be to assume that they represent new molecules in the zebrafish that perhaps no one has ever reported or discovered before. These are novel, perhaps, molecules that aren't in biochemistry textbooks. They're not on Google. They're not in metabolomics databases. And that's certainly possible. But before you jump to that conclusion, it's important to assess some other possibilities by which these signals might correspond. One possibility is artifacts. When you detect signals in an LC-MS-based metabolomics experiment, sometimes what ends up happening is that the baseline will jump up a little. It'll just be a little bit of noise, a little bit of an uptick in the baseline. That can be recognized by the software as a low intensity signal. Okay, but it actually may not be. It just may be a small uptick in the baseline. Another reason that you can end up with signals in the data that you can't identify as a biological metabolite is that they correspond to contaminants. Now, unlike artifacts, which are signals that show up by the software but don't actually correspond to any chemicals, contaminants do correspond to chemicals, but they are not chemicals that are originating from the zebrafish. So what I'm showing you here is a chemical known as an alkylamine. And you can see it has a really strong signal. It goes way up. And that is a common impurity that we encounter in our solvents when we do liquid chromatography. And you can see that it ends up showing up in the experiment. So you have to make sure that your signals aren't contaminants. And then lastly, one metabolite can actually show up as multiple different signals. The metabolite can break into pieces. I'm calling the metabolite in this case E. The metabolite can break into different pieces, as shown here, smaller pieces, or the metabolite can form adducts with various salts, like perhaps sodium salts. Okay, and that can cause one metabolite to show up as multiple different signals. So when we think about metabolomics experiments and metabolomics data that we generate from an organism like a zebrafish, it's good to keep in mind a roadmap for the data. So what I'd like to do in the next couple of minutes is provide you with what, in my mind, is a nice roadmap of the structure and organization and really reflects the underlying complexity of metabolomics when you're analyzing something like zebrafish. So the first thing that you have to recognize is that some signals will be of biological origin, meaning that they're coming from the zebrafish, and some signals will not be of biological origin. Okay, and those signals that aren't of biological origin, as we just said, can be contaminants or artifacts. Again, contaminants being things like impurities, plasticizers from the tubes that you're using, could also be carryover from a prior experiment. There's a lot of sources of contamination, and we've talked about artifacts. If your signal is in fact biological, meaning that it originates from the biological sample, then there's a couple of different ways that biological signals comes in different flavors. It can be redundant, as we said, meaning one metabolite can show up as multiple things, and that's because it can form sodium adducts, potassium adducts, ammonium adducts, et cetera, et cetera. There's also naturally occurring isotopes. So 1% of all of the carbon in the universe is C13. That C13 gets incorporated into metabolites, and because we're using a mass spectrometer and C13 has a different mass than the rest of the carbon in the universe, which is C12, that can lead to multiple peaks. We also have oligomers, meaning that metabolites can form complexes or interact or form dimers with each other, or even tetramers, or et cetera, more complex interactions. And then we have fragments when a metabolite can break into multiple pieces as it enters a mass spectrometer. But what we all hope for and what we are most interested in are those metabolites that are unique. And of course, those metabolites that are unique will have those that are identified and those that are unidentified. So really the million-dollar question is, what frequency do we see all of these different types of signals in a mass spectrometry-based metabolomic data set? And what I'm showing you on the far right there is a histogram that is meant to denote the relative frequency of these types of signals in samples like zebrafish that we encounter in our lab. Okay, I will mention that some of these are going to be specific to certain workflows, like the contaminants that we see in my lab when we do metabolomics may not look like the same contaminants that you see in your lab if you're using different solvents or plastics or things like that. So these do have different specificity, but I will tell you that other labs have also rigorously went through and tried to completely annotate all of these signals. And in general, the same sorts of results have been observed, at least categorically, among different places doing the same kinds of experiments. And I'll just draw your attention to the relatively small fraction of signals that we detect in a standard metabolomics experiment that are really pertinent to biology. Those signals that are of biological origin and that are unique. And you can see it's just the small amount that I've depicted in the histogram there is bright green. And so if we go back to this data that I showed you earlier, where I show you this pie chart, and I say there are 25,000 different signals that we're detecting in zebrafish, many of which we can't identify, it's really important to recognize the vast majority of those signals, at least in most experiments, do not correspond to unique metabolites that are biologically relevant. Okay, I've labeled them noise there. They're not noise in the traditional sense, but they're noise in the sense that they don't correspond to unique metabolites that are originating, in this case, from the zebrafish. Something like only 5% of the signals. So that's really important to keep in mind when you're doing these kinds of experiments, because you don't want to waste a lot of time on unknowns. I call it here the unknown distraction. You don't want to waste a lot of time on signals that don't correspond to anything that's meaningful. So that's just a little background on metabolomics in general. And what I now like to do is say a few words about some methods that we've developed specifically for various experiments that we've been performing on zebrafish. So I talked earlier about doing analysis on entire zebrafish. You can, of course, do metabolomics on entire zebrafish, and that certainly can be insightful. But it can also be insightful to try to analyze different parts of the animal. And what I mean by this is, if you're going to do metabolic physiology, then you'd like to know which metabolites are coming from the liver, which metabolites are in the blood, which metabolites are in muscle, the brain, etc. Okay, we want to be able to make those types of differentiations. Now, zebrafish are relatively small compared to other model organisms that are often analyzed by metabolomics, like, say, mice or rats or humans. And so it does require some development to try to make sure that you have enough material to do the experiments. What we found in our lab to be the best way to analyze serum is to amputate the caudal fin of the animal and then put it in a tube and rapidly spin it down for a short period of time so that we can collect the blood. And we find in our hands that this gives us about twice as much volume of serum for doing metabolomics compared to, say, if you try to do a direct pipetting technique where you try to pipette the blood out of the animal. Okay, and this is important. That twofold increase is really important because, as it turns out, that really puts us above the threshold of what allows us to get really robust, high-quality metabolomics data with mass spectrometry. Additionally, we found that when you're doing metabolomics, you're, for example, extracting tissues or trying to analyze the animals, we find that it's better to use cooling to anesthetize the animal rather than using the more conventional method, which is Tricane. And you can see that, for example, when I'm plotting glucose levels there, I could show you other metabolites as well, that we get much tighter clustering if we use cooling as an anesthetic as opposed to Tricane. You can see that the distribution of the data are much narrower. And as I mentioned, in other cases, you might want to analyze tissues. I'm not going to go through this in detail today, but we've gone through a lot of different isolation methods. There are a lot of different protocols. There are literally thousands and thousands of protocols online for performing metabolomics with mass spectrometry. And so you have to think about which of those techniques are going to be best suited to doing zebrafish metabolomics. And so what we did here, I'm showing you just a heat map to give you a flavor of how we've done this, but we tested a handful of different techniques for isolating different metabolites and compared them in different tissues to see, say, on something like 1,000 metabolites, how much of the metabolite we can collect from each of the different tissues with the different extraction methods. And what this allows us to do is to not only analyze the zebrafish intact, not only analyze metabolites that are circulating in the serum, but also anatomically dissect key organs like the liver, the intestine, the heart, muscle, eye, fin, and brain I've listed there, and then actually evaluate those separately from individual animals. So we're not pooling multiple tissues together. We're getting these from individual animals. And then we isolate the metabolites from those dissected tissues. And what we can do is not only measure things that are changing, but we can get a sense of how much metabolite is in each of those tissues. What I'm actually plotting here is a drug that we administered to this particular animal, and it gives you a sense of where this drug distribution is in the animal after treatment. Now, of course, that's one way of doing these experiments, is taking the animals and anatomically dissecting them. That's somewhat of a complicated and work-intensive process to be able to isolate each of those tissues and then evaluate them separately by using LC-MS. And so what I'd like to do here is present an alternative method that we've been applying to study zebrafish in some contexts, and that's when we use mass spectrometry-based imaging. So it's just like metabolomics that I explained to you before, where you're using LC-MS. Remember, we said that was liquid chromatography mass spectrometry. But instead of using LC-MS, we're using MS imaging. Okay, so you could call it metabolomics imaging. And let me just give you a quick preview of how this experimental technique works. So what we have here is effectively what is a microscope slide. Okay, then we take a zebrafish and we take a cross section of that zebrafish and we deposit it upon that microscope slide. Then we put that microscope slide with the zebrafish tissue on it inside of the mass spectrometer. And then we take a laser and the laser irradiates the animal. And you can see an incident laser coming in here and hitting one part of the zebrafish. Now, when that laser hits the zebrafish tissue, it causes the metabolites to go into the gas phase, and then we can detect them with the mass spectrometer. So what I'm plotting here are a bunch of signals that are originating from the tissue, specifically from the region of tissue that was hit by the laser. And so what you can do is you can take that laser and you can hit one spot of the tissue and you can get a mass spectrum. And then you can move the laser over to the next cell and you can hit that spot of tissue and you can get a mass spectrum. And then you can move it again. You can systematically raster that laser across the entire tissue and you generate a mass spectrum at each location. And then what you can do is you can take your favorite metabolite, whatever one of these peaks corresponds to your favorite metabolite, glucose, amino acids, cholesterol, etc., and you can plot the intensity of that metabolite as a function of the anatomical location within the animal. And so let me just give you a couple of pictures of what that looks like. This is a picture of a metabolite, a phosphatidylserine. You can see that it's really concentrated up here in the central nervous system. This is a picture of histidine, which really illuminates here by the spinal column. Here's a picture of lactate. It's a little more intense here in the under region, and we have inosine here, which is more high in the back there, in the fin. So this gives us a way. Of course, these are just 4 metabolites. These aren't all of the metabolites in the animal, but these are just 4 representative examples of how you can go about interrogating where these metabolites are in an animal. Now, there's one last method that I want to mention to you before I get on to one example, and then my time will be up. But before we get into that example, I'd like to talk about one other method that's related to metabolism, and it's a flavor, a different kind of metabolomics technique. Okay, and to motivate that, I like to show you this picture here. This is a picture that was taken from a bridge above a highway. And it's just a snapshot. You can imagine you have your iPhone and you go stand on a pedestrian bridge above the highway and you take this shot. Okay, now if I shared this slide with you, if I shared this image and I asked the audience, and obviously we can't do this today, but if I could ask the audience, if we were in person and I could ask the audience whether or not what's going on here in this picture, I suspect about half of you would say that those cars are sitting stationary because they're not moving. You know, maybe it's rush hour traffic. Maybe there's an accident somewhere up here on the highway, and all of those cars are just sitting there waiting, hoping to get home and commute through this, you know, get past the accident. But I suspect about half of you would say, no, those cars are moving. They're buzzing down the highway. It's congested. Maybe it's rush hour, but they're all traveling, you know, at the speed limit, going, you know, really fast down the highway. And the point is that you really can't tell by looking at this picture which of those 2 situations it is. And they're very different situations, right? In one situation, the car isn't moving at all. 0, they're not moving, no velocity. And in the other situation, the cars are moving very rapidly at maximum speed. So you have 0 speed and maximum speed. And with just this single photograph, we can't tell the difference. Now that's relevant because when you do metabolomics in the way that I've presented to you already, where you're just measuring concentrations of metabolites, it's equivalent to taking a snapshot picture of metabolism. It's like standing on the pedestrian bridge with your iPhone and taking a picture. Okay, what we actually need to do if you want to try to sort out those 2 examples is we have to use stable isotope tracers. Okay, so this is an area of metabolomics that you might hear referred to as isotope tracer analysis or fluxomics or metabolic flux analysis. But the idea is that you give an animal a nutrient of some kind, and it's isotopically labeled, so it looks different than any other nutrients. And then you watch with time how that labeled nutrient gets propagated through metabolism. Okay, so what I'm showing you here is a bunch of metabolic pathways. Each of these arrows represents a transformation in metabolites. So you can imagine glucose comes in, it gets transformed to glucose 6-phos, right? All these things are different intermediates. And A is the nutrient. Well, let's imagine that we isotopically label A, so now it's colored blue. Okay, and we give that to our zebrafish. We can watch with time how that blue label propagates through metabolism and starts to show up in other metabolites. Okay, and we can map which metabolites it shows up in and how quickly those metabolites get labeled. Okay, and that's now like not standing on the pedestrian bridge and taking a picture with your iPhone, but actually taking a movie of metabolism. So it gives you a whole temporal time course and allows you to understand metabolic kinetics in ways that metabolomics without any labels does not permit. So the nice thing about doing zebrafish, and this is what I think is really amazing, is that it's very convenient to do isotope tracing in zebrafish. If you're trying to do these types of experiments in something like a mouse, it can be somewhat complicated and tedious. The most common way that we do this in mice is you would surgically insert a catheter into a mouse, and then you infuse the isotope tracer. Okay, but it requires a surgery, and it's stressful for the animals because they've had to undergo this surgical procedure. But what we can do with zebrafish is we can simply put the isotope tracer in the water. So I'm showing you here a zebrafish that's in a beaker, and the brown stuff here on top of the water is meant to depict labeled glucose. Okay, and what you can see is if we just look at the amount of labeled glucose in the zebrafish over time, this is hours on the x-axis, and this is how much of the glucose is labeled, you can see that it increases and then reaches a steady state value. Okay, and we can do this easily in zebrafish. And what's nice is you might say, well, what happens if you add all this glucose to the water? Does that affect the animal? It doesn't. You can see that even though we've added the glucose to the animal, the level of glucose in the animal, in the serum, doesn't change. And actually no metabolites in the zebrafish change after we've exposed them to this high level of glucose either in the water. So the water itself doesn't change metabolism. It's a very non-invasive, simple, straightforward way to introduce isotope tracers in a model organism. I really think that this is a huge strength of zebrafish compared to other model systems, the simplicity and the convenience by which we can do these experiments. It's really transformed some of the isotope tracer analyses, metabolic flux experiments that we've been doing in our lab. It's really quite astonishing. So what I'd like to do in the last couple of minutes, I think I just have a couple of minutes left, is I'd like to now talk about the application of these technologies in a very simple context to help us answer a question. I've told you a lot about how we can do metabolomics in zebrafish, but now I'd like to tell you what it looks like in action. And what I'm going to talk about today is the application of these technologies to study cancer, and specifically a certain type of skin cancer that probably everyone is familiar with, known as melanoma. And to those zebrafish aficionados, you know that this is a really well-established model for melanoma. It's a great model in zebrafish that came out of the Zahn lab, the BRAF V600E melanoma. What I'm showing you here is a zebrafish that has developed a melanoma tumor. So this black, ugly-looking thing here is a melanoma. This is a genetic model. So this mutation in BRAF is put into the melanocytes in P53 deficient animals. And then into the adulthood, these animals spontaneously develop these tumors. And we complement these analyses with studies of melanoma in mice and human patients as well. But what I'm going to do today is just focus on our experiments in zebrafish. But I'll just tell you that everything we've discovered in zebrafish is very consistent with what we're finding in human and well as other models like mice. So the first thing that I want to do is tell you a little bit about one of the unique metabolic attributes of melanoma tumors. And to establish that unique attribute of melanoma tumors, I want to show you this screenshot here, which is a PET image of a human who has a tumor. This person had melanoma, and it metastasized to the nasal fossa, the lungs, the lymph nodes. You can see the liver, the peritoneal cavity, maybe a little bit of the vertebrae there. So those are all metastasized tumors. And what we see with these tumors is that when we give patients, this is a human patient, fluorodeoxyglucose, and we look at them with a PET scan, that they take up a lot of glucose. So this PET scan is really just to show you that there's high glucose uptake everywhere the tumors are. That's one of the hallmarks of cancer, is that they have a high avidity for glucose. They take up glucose faster, tumors do, than most of the other healthy tissues in the body. So one of the first questions that we can ask is, do melanoma tumors in zebrafish behave metabolically the same way? That is, do they also take up more glucose than the other healthy tissues in the zebrafish? And the answer is yes. This is an image. It's not quite as fancy as on the left, which is a PET scan. We did this experiment with metabolomics, mass spectrometry, but we administered deoxyglucose, which is a proxy for glucose. And we looked at the uptake of that compound in all the different tissues. And you can see, just like on the left, the uptake of that glucose molecule by the tumor is 10, 20 times higher than any of the other healthy tissues. So that aspect of melanoma is conserved in zebrafish and in humans. So the next question is, okay, we've got these tumors, And these tumors are pathological, and they're taking up all of this glucose. So they're taking up glucose at rates that is much faster than all the other healthy tissues. So you probably know that glucose levels are, it's important to have glucose homeostasis, right? You don't want your glucose to get too high, you're hyperglycemic. You don't want your glucose to get too low, you're hypoglycemic. It's very carefully regulated. Okay, and when it's not regulated, you get diseases like diabetes. So if you have these pathological tissues and they're taking up all of this, glucose, how does that influence glucose homeostasis in the animal? Well, we can go in and look at the serum, right? We talked about how we can do metabolomics on the serum. And what you find is that even though the zebrafish have tumors, they're taking up a lot of glucose, that the level of glucose in the serum does not change. Glucose homeostasis is maintained, which is very interesting. Now, the same cannot be said of every metabolite in the zebrafish. Okay, and what I'm showing you here is a heat map of different metabolites. These are 50 different metabolites. Each row here represents a different metabolite. And on the left, we have the levels of those metabolites in tumor animals. And on the right, we have the levels of those metabolites in wild-type animals. Okay, and I'm comparing liver, intestine, eye, brain, and muscle of these 50 metabolites or so. Okay, and what you can see is that even though the tumor is on the skin, remember this is melanoma. Melanoma resides on the skin in the melanocyte lineage. Even though that's where the tumor is, you can see that there are dramatic differences in the metabolism of the liver, the intestine, the eye, the brain, and the muscle. Okay, this probably, in my opinion, is the most interesting slide that I'll show you today, because what it highlights is that, you've got a tumor in an animal, and we know, people have thought about how tumor metabolism is dysregulated for a long time, but it's really striking that not only does the metabolism of the tumor or the cancer cells get weird when you have the disease, when you have malignant tissues, but a lot of the other healthy tissues respond metabolically as well. And so we want to try to understand what all of those changes mean. And we think the zebrafish is an optimal organism to try to make that kind of assessment. And what I'm going to do here in the last just couple of minutes that I think I have about 5 minutes left. What I'd like to do is just give you a brief preview of one of the stories that we've been able to work out to try to explain why metabolites are changing when you have a tumor in some of these other tissues. That is to say, you've got a melanoma on the skin, and why does it change the metabolism of the liver? Okay, and I'd like to spend a little time walking you through what we found. Okay, so what we have here is a tumor, and we know, because I've already told you, that the tumors in these zebrafish take up a lot of glucose. Okay, where does the glucose go? Well, the glucose gets metabolized through a pathway called glycolysis to pyruvate and lactate. Okay, and this is a very well-established phenomena in tumor. Remember, I told you that tumor hallmark of cancer metabolism is they have a high avidity for glucose. And most of that glucose is transformed into lactate, as shown here. But what we find that's very interesting in these zebrafish melanoma is that a lot of the glucose doesn't get transformed just to lactate. It also produces alanine. Alanine, pyruvate is the keto acid of alanine. The only difference between pyruvate and alanine is nitrogen. So you add a nitrogen to the alanine, to the pyruvate rather, and you get alanine. Okay, then this alanine is produced in the tumors. Now, this is very unique, as I said, because the so-called Warburg effect, which probably many of you have heard, says that glucose gets transformed mostly into lactate. And we obviously see that, but we see a lot of this glucose carbon making alanine. And in fact, it happens so extensively that it's much more prominent. Glucose to alanine transformation is even more prominent in the tumor compared to muscle. And we know in the muscle that muscle is a tissue that takes a lot of glucose and makes alanine, but we get twice as much of that in the tumor. So it's really quite striking. Now that alanine gets made in the tumor, but it gets released into the blood. And we can watch all of this because remember, we can label glucose. We can watch glucose make pyruvate. We can watch pyruvate make alanine in the tumor. And then we can watch that alanine get released into the blood. And then we can watch where the alanine goes. Okay, so where does the alanine go? The alanine goes to the liver. It gets taken up by the liver. The liver takes off the nitrogen and throws that nitrogen away. It actually releases it into the water. And if we measure just nitrogen waste in the water, urea, actually we see an accumulation in urea in animals that have tumors by about 2 to 3-fold. Okay, and that produces pyruvate in the liver. And then what pyruvate does is it goes back through the pathway that we saw over here in the tumor, but instead of going in the down direction, it goes in the up direction. Okay, that's not glycolysis. That's instead a pathway known as gluconeogenesis. Okay, and that's a known pathway. Usually it only happens in our bodies when we're fasting, when we're short on glucose. It's one way that when you don't eat for a while, your body can respond because your liver can make glucose to feed your brain. Okay, these animals are fed, but because they have tumors, they're running their metabolism in what looks a lot like a fasting way. That glucose made by the liver is then released. Remember, we can watch this. It goes to the blood, and then it gets taken back up by the tumor. So what we have here is a metabolic cycle between the tumor and the liver. The tumor takes up a lot of glucose. It converts it into a waste product in the form of alanine. Alanine gets shipped and cargoed to the liver, and then the liver works some magic and takes what the tumor thought was trash and reconverts it back into gold or nutrients and sends it back to the tumor. Okay, so it's a cycle here that allows the tumor and the liver to function together in a kind of a symbiotic way. Now, you may wonder where that nitrogen comes from. Well, we found that nitrogen comes from branch chain amino acids, which are essential amino acids. And you can see that in zebrafish, there's an enzyme that's required to use branch chain amino acids known as BCAT1. And you can see that it's essentially non-expressed in melanocytes, but once the melanocytes are transformed with this BRAF V600E, it's highly activated. And we see the same pattern in human tumors, human melanoma as well. Okay, and so these animals are addicted to the tumors are addicted to branch chain amino acids. They bring the branch chain amino acids in, that generates the nitrogen. They rip off the nitrogen, and then they got to get rid of the nitrogen. And so they do that by sticking it on pyruvate and sending it to the liver so they can be cleared from the animal. Now, the last idea that I'll leave you with here today is what happens if we can block this cycle. Can we go in and use a drug to pharmaceutically block the ability of the liver to reconvert alanine back into a nutrient for the tumor? And if we can do that, would that prove to be an effective therapeutic for treating melanoma? So there's a drug that we can use to do that. It's called Alt-I, and it blocks the enzyme that takes alanine and reconverts it to pyruvate, as I've shown you over there in that slide. And so what happens if we give animals that have these tumors, these BRAF tumors, Alt-I? Well, if you give it to them for 10 days, as you can see here, in just 10 days, we get a dramatic reduction in tumor size. Okay, we get about a 50% reduction in the burden, in the tumor burden, after 10 days of administering these animals Alt-I. So we're really excited about this possibility as potentially a novel therapeutic approach to think about treating melanoma. And if you'd like to learn more about this, we published this paper last year detailing this story and cell metabolism. And I encourage you to go and look the story up. So with that, I'd like to wrap up. I've hopefully given you a little bit of context for how we perform metabolomics in our lab and given you a short vignette of one example by which we've applied it to study melanoma and zebrafish. With that, I'd like to thank everyone in the lab who was responsible for making this work possible, in particular, Maddie Jackstad, who I already showed you on the first slide, and one of her partners in crime, Fuad Nasser, who's pictured here. That's Maddie on the far right here, and this is her partner in crime, Fuad, right next to her. As well as everyone else in the lab, I'd also like to thank my mentor, who we tragically lost a couple of years ago, Steve Johnson. He taught me so much about zebrafish and inspired me and drew me into this model organism, and it's been a really fun adventure, and we are continuing to learn a lot of really exciting science. So I'd like to thank him for all of the information and collaboration that he taught me over the years. And thank you for your time and your attention. Thank you.
So thank you very, very much, Professor Gary Petty. We're very pleased to have you here. Very nice work. Very different, actually, because I've never seen something like that. We actually don't have any questions from the audience until now. I don't know if anyone wants to ask something right now. Last chance, everybody. But If not, they maybe send something in the comments watching the video later, and then we can forward the question to you later.
Absolutely.
Well, I guess no one's going to make any questions, so that's it. Thank you very much, Professor Petty.
Wonderful.
We're very pleased to have you here.
Thank you.
And we hope to see you again soon.
Thank you. Thanks, everyone. Have a great day.
Great day. So I'd like to invite here to get along with me all my friends from the organizer committee. Ives, Renata, Ana, Flávio, Jorge, everybody. Can you hear me?
Yes.
Yes, yes.
I think they're all here now. I don't know if there's anyone missing. Oh, Monica. So first of all, I'd like to thank everybody from the organizer committee, especially Ivy, who's the idealizer of this webinar, but everybody worked. So thank you very much for everyone. And obviously, thank you very much for your time and your presence here to see us, to hear from us, and also listen all these speeches from these speakers, marvelous speakers from around the world. And it's a great pleasure for me to be here. And I guess I'm speaking for everybody when I'm saying this, but I think all of my colleagues are very happy with this webinar. And I'd like also to thank all the participants, the speakers, the marvelous speakers that we had this week. It was more than 30 hours of live streaming and a lot of hard work in the background and the back part of the webinar. So thank you very, very much to everybody. If anyone wants to say anything now, please do. Don't let me.
Thank you, guys. See you next year. Renata? Jorge?
Yes. Yes, well, just only to acknowledge the participation in this excellent event. So then, as we saw, there were excellent talkings. It's top-notch researchers from all around the world, and then it's quite obvious that the Zebrafish, it's a very powerful model, and then we should keep going on our research. And then I think that the effort that particularly Yves paid on this event was wonderful. So then I hope to see you next year. Thank you very much.
And actually, there's another person that I forgot to thank, but it's the probably one of the people that deserves the most thank you ever. That's Marcio from the ICB, because he's the one working with all the things related to the streaming and the live. So thank you very much, Marcio.
Flávio, do you?
Yeah, well, I want to say thanks for allowing me to participate in this event. It was really, really wonderful. I really enjoyed all the talks I was able to be at. It was really long and tiring. I guess you, the ones of you who were really working all along, because I cannot say I was. I'm not really organizing. I just was helping a little bit with very minor things. So, but anyway, I thank you for letting me to participate in this, and congratulate all the participants, both the speakers and people who was present at the talks while the talks were being done. Of course, there will be people looking at them later, but the nice thing is when people is there and there can be discussions, and that was. Thank you for all of you who were there.
Ana?
Well, I want to thank you too, mainly you, Ives, that was the leader of this organization here and could organize everything and invite everybody, very nice speakers and all these people that are behind all this webinar. So thank you everybody for being here. We know next year we have in person a conference in Minas Gerais, so everybody's invited to there. It's going to be in Brazil, so you, anywhere you are, so come to Brazil and see us there. See you.
Mônica.
So I just want to thank you for the opportunity to participate from something so big. And we know that it's big because everyone is saying that. So Yves, congratulations. I think that you were the most important people from this event because you fight a lot to do this. And thank you to give me the opportunity to participate from it. Thank you.
Leonardo.
Thank you, Yves. Congratulations. Good job. Next year, a new event in bigger and small, in person or online. See you next year.
Yeah. Thank you, guys. Obrigado, pessoal. Cada um na sua língua agora.
Valeu,
Gracias. Um prazer.
Tchau, tchau.
Obrigada, gente. Tchau para vocês.
Até o próximo ano.
Thank you, Ives.
Tchau, tchau.
Bye bye.